We will now be talking about chromium. Chromium is a trace mineral found in a variety of plant and animal-based foods, including eggs, beef, poultry, grains, cheese, and dark chocolate. While its method of digestion and absorption is not fully understood, there is preliminary evidence linking chromium to improvements in the regulation of blood glucose. You will see in this slide presentation how chromodulin, which is a complex consisting of 4 chromium ions, binds to the cytosolic beta subunit of the insulin receptor and stimulates the kinase activity of the insulin receptor, which facilitates insulin's role in regulating blood glucose. Chromium exhibits several parallels and interactions with iron. Therefore, high-dose supplementation of chromium may affect an individual's iron status. Lastly, we will review the DRI values for chromium and its deficiency symptoms.
Here we see food sources of chromium. We see that is in food. It is present in the trivalent form, so there are a number of oxidation states of chromium ranging from chromium 2- to chromium 6+. However, in food it is seen in this trivalent form. We see that chromium is found in a number of food sources, so we see beef and fish and poultry it is in, so these animal sources, also cheese, and the plant-based sources would be whole grains as well as peanuts. We also see chromium found in dark chocolate, different fruits and vegetables such as green peppers, mushrooms, apples, and bananas. Additionally, some spices and brewer's yeast. We'll discuss the absorption process for chromium. We see that it is released from food in acidic solutions, so in an acidic environment such as the stomach, we see that the chromium mineral is released from food, so then we have the free chromium available to be absorbed in the small intestine, and primarily that is occurring at the level of the jejunum. We see that the mode of absorption is either passive diffusion or via a carrier-mediated transporter, though we won't go over this process in detail, but just to know that it's either the passive diffusion or a carrier-mediated process. We see that the percent of chromium absorption is very low, so it's about 0.4 to 2.5%, so it's very low, similar to the non-heme iron. We saw that the percent absorption for non-heme iron was about 2 to 3%. And so that rate of absorption is similar with chromium. We'll now discuss the transport of chromium through circulation. So like iron as well, we see that the chromium will bind to transferrin in blood to be transported in the bloodstream. So again, like iron, chromium does also bind to transferrin. It also binds to albumin. And so this protein albumin will also carry chromium through circulation. Similar to iron, we see that chromium is stored with ferric iron. So in the storage site, it's going to be similar as well to iron. And it can be concentrated in storage in the kidney, liver, muscle, spleen, heart, pancreas, and bone. So we do see a lot of these parallels and overlap between the chromium and iron. So a key function of chromium that has been recognized in the literature is that it facilitates the action of insulin. So when we have insulin binding to insulin receptor, that will promote a signaling cascade within the cell, which then leads to a series of events and a number of outcomes within the cell. So a key outcome that we've discussed previously is the translocation of GLUT4 channels to the cell membrane. So a outcome or a result of insulin binding to the insulin receptor. So it initially will promote a signaling cascade within the cell, which then will lead to the translocation of GLUT4 channels to the cell membrane. So the chromium will actually facilitate this process, will facilitate this intracellular signaling cascade and then the outcomes of the insulin binding to the insulin receptor. And we also see that chromodulin, it's a chromium, a functional form of chromium, and the chromodulin is synthesized in response to the secretion of insulin. We see that within the cell, it binds to the beta subunit of the insulin receptor, stimulating the kinase activity of the subunit of this beta subunit. So the chromium will bind to this beta subunit, then thus stimulating the kinase activity of this beta subunit. With the binding to the beta subunit, it promotes and facilitates the intracellular signaling cascade occurring in response to insulin binding to the insulin receptor, thus promoting an increase in this GLUT4 translocation. So it facilitates the effects that occur when insulin binds to the insulin receptor. Therefore, it will play a role in glucose and lipid metabolism. We see here the diagram here of a cell. We have either a muscle or adipose cell. For example, we see the transferrin bound to chromium binding to the transferrin receptor, and we see that this is taken into the cell. We see that the chromium is released from the transferrin receptor. The chromium released from the transferrin receptor binds to this apochromodulin, and this apochromodulin binds to the 4 chromium ions, then forming chromodulin. So chromodulin is apochromodulin bound to the 4 chromium ions, and then this chromodulin structure then will bind to the beta subunit of the insulin receptor. So we see this is the beta subunit particularly of this insulin receptor, and we see that this is occurring with the binding of insulin to the insulin receptor. Chromodulin binds the beta subunit, which then enhances and stimulates the kinase activity of the insulin receptor. So as a result, there have been several research studies investigating the effect of chromium on different disease states and conditions that are related to insulin activity. So one would be diabetes. Also, we see studies looking at the role of chromium in regulating body weight and body composition. So the initial studies that have been done evaluating the role of chromium in diabetes, we see that there are preliminary results documenting a beneficial effect of chromium supplementation on hemoglobin A1C, and so this is a marker of indicating blood glucose regulation. So if individuals have adequate regulation of blood glucose and do not have elevated glucose levels over a prolonged period of time, then hemoglobin A1C will be in the normal range. However, if this is elevated, then it indicates an overall more of a long-term elevation of blood glucose levels. So chromium supplementation had a beneficial effect on the hemoglobin A1C levels, as well as fasting glucose in individuals with type 2 diabetes. So this is promising. It is preliminary evidence, and there have been some studies with conflicting results, so some inconsistent results. However, we do see some preliminary evidence of this beneficial role of chromium supplementation on blood glucose regulation and hemoglobin A1C levels. There are some dietary supplements containing chromium that have claims for its aid in the regulation of body weight and body composition. However, claims have not been validated or supported by research. So while we do see these products available, the evidence supporting its role as it relates to weight loss, changes in body composition, increasing in strength has not been substantiated by research. We have seen relationship and parallels between chromium and iron. So we do see, as a result, some interactions with iron. So chromium is transported in the blood bound to transferrin, However, the effect of chromium bound to transferrin on iron's ability to bind to transferrin is unclear at this point. So the potential for chromium to displace iron in transferrin is unclear at this point. However, it may affect the iron binding to transferrin potentially. We see that chromium supplementation led to decreased serum ferritin, total iron binding capacity, and transferrin saturation. So there may be some preliminary evidence here suggesting an effect of chromium on iron's ability to bind transferrin. We see the AI level for chromium for adults less than 50. It's 35 micrograms per day in men and 25 micrograms per day in women. The AI level changes for those older than 50, so in men it's slightly lower, and women also it's slightly lower at 30 micrograms and 20 micrograms per day. In pregnancy, we see it's slightly higher. For women, it's 30 micrograms per day. In lactation, it's 45 micrograms per day. We'll see here deficiency symptoms. So with chronic low intake of chromium and low chromium status, we see various deficiency symptoms such as weight loss, peripheral neuropathy, insulin resistance. So this is going to be a key one to focus on as it relates to chromium deficiency, as well as high plasma free fatty acids. There is no tolerable upper intake level for chromium.